Okay, let's start this video with a somewhat intriguing story, or I guess a somewhat intriguing medical mystery from 1980s. And here the story starts in Papua New Guinea. Here, deep in the remote mountains, lived a very close-knit tribe referred to as Fore, a tribe that had a somewhat heart-wrenching tradition. When one of the loved ones or a family member died, Fore believed that consuming their body was a sacred ritual meant to free the spirit and keep their essence alive inside the family. So essentially they were cannibals. And in this ritual, women and children played a special role. Here, by eating the brains of the deceased, which Fore believed seated the person's soul and person's wisdom, they would essentially be blessed by the spirit of this person, and thus give them a kind of a symbolic immortality, or at least have their spirits live inside the whole family. But in the mid-20th century, this custom unleashed a somewhat deadly secret, a terrifying disease referred to as Kuru. And while unknown to them, Kuru was actually caused by something we didn't really understand until the last few decades. A type of a rogue protein referred to as a prion that can be spread from one person to another if consumed directly. And so here this tragic epidemic showed us how a protein can actually pass through generations, becoming a kind of a stealthy killer. But for the medical field, and more widely for the field of biology, this was an incredible discovery. Here the story of the tribe of Fore and their fight against Kuru led to a discovery of something we had no idea even existed. Something that doesn't actually look that special or that interesting. It literally just looks like a typical protein. But in this video we're going to go just a little bit further in exploring this idea of prions and actually tackle one of the most profound questions in biology. The origins of life on planet Earth through the process known as abiogenesis. And so this is actually the next part of our exploration of the origin of life, but this time we're going to focus on this very bizarre protein that even today we actually still know so little about. But first, let's briefly discuss what we've talked about in one of the previous videos in the description, because in some sense all of this actually connects. Here, let's talk about the RNA World Hypothesis that a lot of scientists believe might have formed early life. And this is something that a lot of scientists believed for decades. The idea that RNA, a molecule related to DNA, was very likely the first genetic material in the world and eventually led to the evolution of everything. And that's because in this case it can actually store information pretty easily, but at the same time is also able to catalyze various chemical reactions and has a lot of additional functions that DNA does not have. And so in some sense RNA is a perfect first molecule to possibly start life anywhere, except that maybe there's just one problem. RNA is also notoriously unstable. As a matter of fact, it even falls apart inside water. And so here many scientists struggle to explain how such an essential building block could have formed spontaneously on the planet and could have then evolved into DNA and everything else. And so to a lot of evolutionary biologists, this actually brought them to another concept that kind of sort of made just a little bit more sense. A surprising alternative referred to as the protein first world. The idea that maybe it was really not the RNA but proteins that appeared first, and maybe it was proteins that eventually produced RNA, DNA, and so on. But at the very heart of this idea, there is something really interesting. A somewhat misunderstood molecule that we're going to be focusing on now, that up until recently was mostly known for causing fatal diseases, the enigmatic prion. Although there's a slight chance that some people might also pronounce it as prion. But when I went to school it was pronounced prion, so I'm gonna stick with that. And so here this is a story of a rogue protein that might hold the key to the life's origin, in essence changing our entire understanding of biology and evolution. But to understand why prions are so important, we really have to understand exactly what they are and how they form. First, what exactly does it mean? Well, it's actually a kind of a shortened form of proteinaceous infectious particle, because that's what we initially thought they were. And the most famous example of this particle that most of you probably heard of is, of course, the mad cow disease, more officially known as bovine spongiform encephalopathy. And I dare you to say this super fast 3 times. I've been trying this for like 5 minutes now. And so here this is a somewhat infamous class of various brain diseases that can be easily transmitted from one species to another and always involve these somewhat bizarre proteins. But the main discovery about prions was made back in the 1980s. Here Stanley Prusiner essentially made the first proposition and potential explanation involving these somewhat misshapen proteins. But at first, almost no one believed them. except that Prusiner was able to demonstrate that infectious agents for pretty much most of these diseases contained no DNA and no RNA, yet somehow these agents were able to spread from one species to the other species and would infect anyone that consumed the flesh. And so he proposed that it was protein, and eventually this was proven to be correct. Which was actually revolutionary because traditionally, any kind of an infection usually involves viruses or bacteria that always rely on genes and replication in order to spread from one organism to another. So basically here we had some kind of an infection that didn't involve any genetic material. And eventually he received the Nobel Prize for this absolutely incredible discovery. And around the same time, scientists were finally able to explain how they actually work. In essence, it is just a protein, but a protein that has been folded into the wrong shape. So just as a side note, in order for a protein to be functional, it has to first form a three-dimensional shape that always has to be pretty much the same, but occasionally some protein can become misshapen and will thus lose their original function. In this case though, for some reason these proteins, as they acquire this new shape, instead of becoming useless and eventually broken apart, actually become infectious, as in they can then affect other proteins and misshapen them as well. And so the healthy protein that's referred to as PRPC can sometimes become misshapen and acquire a new form referred to as PRPSC, which becomes infectious and can then spontaneously create even more infectious proteins by directly interacting with them physically and by causing them to basically assume the same shape. So it's kind of like a corrupting template that continuously misshapens everything around it through the process of misfolding. It's sort of demonstrated in this picture right here. And to be more exact, it actually changes the internal structure from mostly flexible coils to somewhat rigid stacked sheets called beta sheets, which essentially transform this protein dramatically. And once it's misfolded, apart from infecting other proteins, it can also do something else. It can actually start clumping and create larger and larger structures. Specifically, it forms these very long thread-like fibers called amyloids, which can then accumulate in tissues and are the main reason why this becomes so dangerous. Basically, they kind of start clogging everything and can easily cause cell death by causing damage inside the cell because they literally start blocking everything. And while for animal species, like humans and of course cows, this tends to create a lot of unusual holes and spongy structures inside the brain tissue, because all of the cells close to the original infection start to slowly die out. Here's actually one of the pictures of the infected cells from one of the samples. But while disease-causing prions are obviously kind of devastating, quite a lot of recent studies unveiled something absolutely mind-blowing. Turns out that this prion-like behavior is actually quite ancient and widespread, and is technically also necessary for a lot of function in a lot of different biological systems. And so now we know that quite a few proteins have this ability to fold into two functionally distinct configurations, and seem to exist in a lot of branches of life, suggesting that these multifold proteins seem to provide life with some kind of an evolutionary advantage. And so here let's discuss some of these non-disease prions, because that's definitely something that's super surprising and a lot more interesting. And specifically these are known as prion-like proteins, and they seem to play quite a few crucial roles in healthy organisms. For example, we know that they seem to be responsible for memory formation. And so going back to even 1970s, scientists discovered that prions seem to be essential to creating long-term memories. And the way this works seems to be pretty simple. In order for a long-term memory to form, the connection between neurons, the synapses, must physically enlarge, which usually requires new protein production. And here scientists discovered that a protein called CPEB seems to act like a prion. When a synapse inside a neuron is simulated, this protein starts to fold into its prion form and then grows into a stable fibril, which activates the production of all of the necessary proteins to permanently change the synapse and make it a lot more efficient. In other words, these prions inside the neuronal synapses seem to dramatically strengthen the connection, which is literally what a long-term memory usually is. And this has been physically confirmed in various experiments using sea slugs and even fruit flies, and has been observed in a lot of different mammals as well. Then, apart from memory, there's also a kind of a role in adaptive evolution. For example, in organisms like yeast, prions also offer a non-genetic way to adapt to various food environments that would be otherwise quite impossible. So for example, when there's a dramatic change in the environment, some of these prion proteins will switch into a different shape, which happens very quickly and quite suddenly, and allows yeast to then exploit completely different type of food. And intriguingly, this new trait is then also passed to the offspring, literally acting as a kind of a non-genetic inheritance, which by the way is really mind-blowing because we've always thought it can only be done through genes. Yet here we have direct evidence that even prions can create these genetic memories across generations. And last but not least, it also seems to have an influence in immunity. Specifically, prion-like molecules seem to be also used by various immune cells, especially when it comes to fighting off viruses. And in this case, prions usually encourage fast delivery of antiviral proteins, and do so very effectively because they can misfold much faster than a cell can produce new proteins. And so based on these discoveries, when it comes to abiogenesis or the formation of life from inorganic molecules, here we have some really important hints. For example, when it comes to the mechanism of cell propagation, we now have direct evidence that these misfolded proteins can simply convert others very easily, which is of course one of the main properties for life itself. And so when any protein with prion-like properties binds to a similar protein with an unstable shape, they then form a highly stable pair through what's known as self-templating. This is also known as self-templating reproduction. But as the structure grows into a fibril and starts to become larger, it can then start to make lots of copies of itself, especially if this whole structure or this fibril fragments, creating individual seeds that can then do this again somewhere else. Which means that prion-like proteins possess all of the necessary functions for the formation of abiogenic life. First of all, they have an actual function like an enzyme. Second of all, they can reproduce by affecting other proteins nearby. And this leads us to an idea referred to as the Protein World Hypothesis. Kind of like the RNA World Hypothesis, but basically using proteins. And so maybe the formation of complex molecules from non-living matter really started with these prions, because they definitely seem to have everything we need. Especially because we know that a lot of them are relatively easy to build. Building blocks of proteins or amino acids are extremely easy to form spontaneously, which was of course the main discovery from the 1950s Miller-Urey experiment that was able to form a lot of amino acids by just putting them in extreme conditions very similar to early Earth. Not to mention that many amino acids are also found on meteorites and in most molecular clouds in outer space. We also know that amino acids generally polymerize or link pretty easily. They can form very long chains of peptides, especially in locations with a lot of wet-dry cycles. And that means that they can also produce some of the simple early proteins. And while by luck, at least some of these proteins might become functional and even act as catalysts for additional chemical reactions. So basically, even some of the early proteins can then start supporting metabolism. But crucially, they're also stable. Unlike RNA, many of these proteins, especially the ones that are prion-like, seem to be extremely resistant to harsh environments, which is why mad cow disease spreads so easily, because even cooking meat in this case would actually still maintain the shape of these proteins, allowing them to spread further. Additionally, some of the earlier experiments even showed that some of these prions can easily survive some of the most extreme conditions, including hydrothermal vents, where today scientists believe life might have started sometimes 4.3 billion years ago. In other words, they seem to contain all of the necessary properties to possibly begin life. Once again, in contrast, the RNA World Hypothesis still faces quite a few challenges, mostly related to stability. But it doesn't mean that the RNA World Hypothesis is incorrect. As a matter of fact, quite the opposite. Today, quite a lot of evolutionary biologists seem to propose that maybe both of these ideas are correct, with most modern research now suggesting that the emergence of life was not just proteins or RNA, it might have been both. And so based on at least some of the studies you can find in a description, scientists are now proposing what's known as the Peptide RNA World, a model emphasizing cooperation between early prion-like molecules and early RNA. Because in this case, it suddenly actually makes a lot more sense. And really because of one thing. We know that proteins and nucleic acids, or RNA and DNA, today are completely interdependent. They essentially produce one another. And that means that they might have cooperated from the beginning and evolved together from early days. But this is not just a proposition. There are actually some experiments. For example, some of the research showed that by using very simple reactions using elements like hydrogen cyanide and hydrogen sulfide, and by applying UV light, we can then spontaneously produce necessary chemical components for RNA and proteins, which basically suggests building blocks of life could have emerged from common origins at the same time, so there's no reason for one of them to come first. Likewise, very short protein chains, referred to as peptides, might have been available much earlier and in higher concentrations than similar complex nucleic acids like RNA. And so in this case, a lot of these early peptides might have acted as a kind of a catalyst in order to accelerate the synthesis of early RNA molecules and to eventually create something that could then self-replicate. Likewise, studies have also shown that short peptides derived from the structure of modern ribosomes, the same machine that then produces proteins, can actually boost the function of RNA enzymes in the lab, suggesting that non-encoded peptides also play an essential cooperative role in supporting some of these nucleic acids like RNA and DNA. Or just to rephrase this, even the ribosome itself, inside of it, seems to contain structures that physically influence RNA by boosting their function, which means that they must have evolved from something much smaller that very likely played a role in early evolution, with the additional experiment involving deep ocean vents showing that the heat flow through various very thin cracks in many underwater locations seemed to concentrate a lot of prebiotic building blocks in such a large amount that it then creates very high concentrations needed to form these polymers and all of these peptides pretty easily and pretty fast. Or once again, just to rephrase this, many of these volcanic locations, such as hydrothermal vents, seem to produce just the right conditions to spontaneously assemble some of these early proteins, which can then start acting on nucleic acids and thus start forming some of the first molecules of life. And so ultimately, here the story eventually ends with the formation of what's known as the last universal common ancestor, something that we've discussed in one of the previous videos in the description, and something that most likely formed approximately 4 billion years ago. This was of course the precursor to modern bacteria and archaea, which possibly suggests that the Luca very likely used quite a lot of prions for its own reproduction, and eventually evolved all of the necessary life functions that we basically take for granted today. As a matter of fact, some things potentially never even changed. We know that ribosome seems to be a pretty ancient structure because it seems to be almost exactly the same in all organisms on the planet. That's once again that protein that's responsible for turning RNA molecules into proteins. But it looks like this collaboration between RNA and proteins was already in existence before ribosomes even became common. And so if we were to summarize this whole timeline, it might look something like this. It all started with some kind of a early prion, which eventually started to collaborate with some of the early RNA molecules. This eventually led to more complexity and at some point became so efficient that it even allowed more complex structures like DNA to form as well and to even become stabilized inside some kind of an early cell membrane. And this was probably the formation of that first common ancestor. And though this probably took lots and lots of trial and error, eventually at least one organism became very efficient. But naturally, right now this is just based on individual experiments and some of the hypothetical propositions, so we're not really going to know exactly how accurate this is until we find something similar somewhere else. So for example, if we do find something very similar on some kind of a moon in the solar system, now then the story is going to become much more interesting. But before we find something, this is still just going to remain a hypothesis. A really intriguing one, and one that seems to explain quite a lot, but not a theory yet. And so we'll definitely come back and discuss this more once there are some updates, but until then, check out previous videos on a similar topic in the description below. Thank you for watching, subscribe, come back tomorrow to learn something else. Support this channel on Patreon where you can find additional videos without any ads and can DM me directly, or by joining a channel membership that grants you early access. You can also support this channel by buying the Wonderful Person t-shirt in the description. Stay wonderful, I'll see you tomorrow, and as always, bye bye.